Next, we will have Yael Balbastre from University College London presenting a scalable computational framework for large-scale imaging of neural circuits.
Thank you. Thank you for coming. Yes, I'm going to present the project we have started, the kind of what we want to do with the data that we acquire and the computational challenges that arise from that type of data, and then the solution that we have been developing for the past year. I have nothing to disclose. So our project builds on two existing challenges. One is large-scale microscopy, so trying to scale very high-resolution imaging of tissue to very large field of views. And there's been quite a few efforts in the last years. And the other challenge is correlative microscopy. So within the same sample, acquire different modalities so that some of them can explain the signal that is visualized in the other ones. So our project, so we have this Center for Large-Scale Imaging of Neural Circuits. And I'm going to start with what our overarching goal is, and it's to build a microscopic scale structural connectome of the human brain. But it's quite complicated because we need to image the data at resolution high enough to follow axons, but also cover the entire human brain. So our strategy is to use both macaque brains and human brains, and critically, both types of brains go through the same imaging pipeline. And in macaques, we also have tracer injection. So we have ground truth pathways from some cortical areas that have been injected down to the entire white matter. And we can then check that the inferences that we make from the microscopy data match the ground truth from the tracer, and then apply the same techniques to human data with confidence. And we don't do it just for the sake of acquiring data. We have use cases. One of them is deep brain stimulation, where having really highly precise connectomes help targeting different cortical tracts that go to different cortical regions. And then we also have another use case, which is to use this really high resolution data paired with diffusion MRI to try to better model the diffusion signal, and again ensure that inferences made using in vivo modeling techniques are valid. So the thing is, maybe I'll go back. So the data we're working with is quite large. It goes from, so our diffusion MRI is writing maybe a few gigabytes, and then the light sheet data for a single slab of tissue is going to weigh several dozens of terabytes. So it's not data that you can really load the way it's usually done in neuroimaging, and so we have to adapt how we store and compute on our data to those constraints. So one is we can't have multiple copies of the data, so everything needs to be centralized into a central repository. In our case, it's on the web. We also need to access very efficiently to different parts of an array of data, and also to different resolutions of data. We need to be able to visualize our data for qualitative control and to check that our processing is correct without having to copy the entire data locally. So this needs to be remote. And we also need to be able to compute distributively to just handle the sheer size of the data. So there are existing tools that have been developed in the last few years. We're using ZAR for data storage, Dandy for the cloud infrastructure. NeuroGlancer is a viewer developed for the EM community that handles remote visualization and then Dask for distributed processing. So ZAR, I just explained in case you're not too familiar, ZAR is a format that handles any mapping between keys and values, and that can be stored on lots of different backends, including on the web. And OME-ZAR is a specification built on ZAR for microscopy data. Then we use Dandy, which is an archive built for neurophysiology data, but that also handles OME-ZAR data and microscopy data. And we're using NeuroGlancer for visualization, which was developed for the connectomics community. And the important point of it is it runs entirely client-side. So the data streams directly from where it is stored into the browser, without going through a third-party server. So it's much more scalable in terms of speed, but also cost of running it. And it's very easy the entire state of the viewer is stored in a JSON object, so we can very easily send one scene to another person, and they'll open the link and see the exact same thing without having to actually share data. There's some limitations. One of them is in this community, there's less focus on anatomy and anatomical spaces. And they are drafting specification for that, but they're quite complicated and take time, and we wanted something that worked quickly and didn't need a lot of talk. So we created this nifty-zar specification that's based on OMEZAR, and we are really just saving a nifty header on top of it in our dataset. And that makes it really easy to operate with existing software. And then NeuroGlancer, doesn't know what anatomy is, doesn't really map XYZ to right anterior superior, and it can load nifties, but that's the only neuroimaging format that it knows, and doesn't know how to handle vector files, which are important for us in terms of connectomics. And like if you open data in Freeview or in any existing neuroimaging visualization software, everything is shown as expected. We know that if it's in the right direction, then our header is fine. If you open the same file in NeuroGlancer, I mean, we can see data, but it's not like we don't know what's happening. It's harder to operate with it. So we created a little Python library that wraps NeuroGlancer and is an interface between your neuroimaging data and the NeuroGlancer scene. So it helps creating NeuroGlancer scenes in a way that mimics what we used to in neuroimaging visualization. And so I'm not showing it, but we also support vector fields as well as geometric transforms. And so now our entire framework, so we acquire these different types of data, diffusion MRI, ex vivo, and then microscopy. Everything gets converted either to nifty-zar for the MRI or to OMEZAR for the microscopy and gets pushed to our archive, data archive. And then using, again, this nifty-zar software, we can very easily access chunks of data but preserve the orientation and then apply classical neuroimaging tools on it, upload our results or transforms, and then apply those transforms for visualization using this ngTools library. So I'm just going to show an example. So this dataset contains a diffusion MRI scan at 800 micron resolution and a hierarchical X-ray phase contrast X-ray that's 10 micron resolution whole brain. And we also have an excised portion of the brain that was re-imaged again at higher resolution. So we can very easily load all of that data, have it coalesced together, and then compare the tracks obtained from the diffusion data to the HIP-CT high-resolution structure. So everything is public and open source. It's under development, so we're adding new features quite often, and we're happy for feedback. So thanks to everyone involved. So that's a very large consortium, and I'm happy to take questions.
Thank you so much. Any questions on the microphone? Don't be shy. Through the app, don't be shy either. I like very much, the approach you have taken that is very modular, bringing together things that already exist and putting some smart glue between those. I guess it's not a question specific to your project, but what was the most, challenging aspect of putting this together in your perspective?
So yeah, I think we were lucky that most of the computational changes themselves had been tackled in EM before, and that helps a lot. So the main challenge is really just to interoperate with it, and it sounds easy, but it takes a lot of development to make sure that everything is interpreted correctly and keeps the performance of the outcomes of the EM, but also the ease of use that we're used to in neuroimaging. So that would be the main one.
That's great. Oh, there's a question. Please go ahead. We have time.
Yeah, there's a question. Thanks a lot for the presentation. I'm wondering, are you aware of a commonly accepted format already for the many streamlines you are showing? I mean, for the voxel data, I know about it, Omizar and so on.
No, so for streamlines, there's nothing that scales as well. It's one of our goals in the next year is to come up with something that scales as well as OMISR for voxel data, and try to agree with other groups that lead to similar projects, I assume including yours maybe. So yeah, it's next year's main objective is to come up for something for streamlines.
Yeah, very interested in that, so maybe we can have a chat.
Of course.
Thanks.
There's a working group I can relay so that everybody can join. Thank you so very much, and congratulations.